Thanks, everyone. You are in the Introduction to Freshwater Mussels session, and I am thrilled to be here to help moderate this session and welcome our speaker, Joe Rathbun. Joe Rathbun is a mussel biologist, among other things. He has a BS in aquatic biology from the Florida Institute of Technology, a master's in environmental chemistry from U of M. He worked as an environmental scientist in the public and private sector for 38 years, specializing in aquatic toxicology, contaminated sediments, dam removals, which I think is how I first met Joe, is when I was working at the Huron River Watershed Council. We were doing a lot of dam work. River geomorphology and restoration, and mussel ecology and surveys. And in his retirement, he's continued to teach mussel identification workshops and assist the DNR, Eagle, and various universities with mussel surveys around the state. If you have questions for Joe as he goes through his presentation, use the chat button to type those questions into the chat. I'll be the moderator, and we'll take breaks throughout the presentation to give Joe a chance to address your questions. But for now, without further ado, I'm going to hand it over to Joe.
Thank you, Joe. Thank you all for attending this session this morning. Mussels are a very interesting fauna. Snails or crayfish, depending on who you talk to, are the most endangered group of organisms, aquatic organisms in the world. The outline of this talk is going to be as follows. We'll have 4 very brief overviews of mussel ecology, survey protocols, mussel identification, and if you want to work with mussels yourself, do your own surveys, do your own IDs, some tips on how to accomplish that. Between each subsection, we will pause for questions. So that's when Joe will read me what you've got in the chat. So there are around 900 species of mussels around the world. There are about 300 in North America. That number is slowly declining. Approximately 10% of the species in North America are extinct, and about 70% of them are on somebody's list as threatened, endangered, special concern, whatever. We have about 42 species in Michigan. I say about because the official list is 42. 3 of them are probably no longer here. They haven't been seen in at least 50 years, but they're still officially on the list. The hotspot for mussels in North America is in the southeast. I'm really glad I don't work in Alabama with 175 species. Mussels are very, very important in the aquatic ecosystem. They are kind of the equivalent of beavers in that they are ecosystem engineers, a term that was first coined for beavers. They improve water quality by filtering the water. They feed and respire by filtering the water. A study from the 1980s looking at the native mussels in Lake St. Clair established that they filtered the entire volume of Lake St. Clair once or twice a month. Their mussel beds are basically like cobble beds in a sense. They're very important for certain fish for spawning areas. They increase the number of aquatic insects by providing good habitat. Like I say, the equivalent of gravel or cobble, and the insects live on the outside of the shells. They stabilize the river bottom, again, like a riffle does. And they also absorb carbon. Their shells are made from calcium carbonate, which they obtain from the water around them, and the water gets it from the atmosphere. And in a dense mussel bed, the shells will accumulate about as much carbon every year as a temperate forest, which is to say around several hundred grams of carbon per square meter per year. They have, like any organism, a variety of ecological needs. They are filter feeders, so they like flowing water, although they certainly do occur sometimes in huge numbers in lakes and reservoirs. They like moderate to high dissolved oxygen. Moderate to low sedimentation. If they're being buried by massive sediment inputs, like say after a dam removal or something like that, or some bad construction project upstream that can kill them. They like moderate pH and calcium, which in most of the state is not a problem at all. We have some very highly buffered waters here in the state. They live on and in the sediments, and therefore they like stable sediments. You don't really find many mussels in urban streams that are hammered with hydrologic flows after a rain event. We'll talk about their reproductive system in a minute, but they do need their host fish species to be present to successfully reproduce. And zebra mussels, they do not do well with zebra mussels, as I'm sure you all know. They have an unusual reproductive cycle. The male and female are separate. They have external fertilization, wherein the male releases sperm into the water column. The female takes it in as she filters for food and dissolved oxygen. That fertilizes the eggs, which she holds seasonally in some specialized gill filaments. I'll show you pictures of this in a minute. And those fertilized eggs develop into a type of larva called a Glochidia larva, which looks like a miniature clam. And those are obligate parasites on fish. They have to spend a few weeks to a few months living on the gills of fish. One species known also uses the mud puppy salamander. So they'll live on the gills of the host fish species for some weeks to some months, depending on water temperature. Then they'll drop off as juveniles and continue the life cycle, growing into adults and reproducing. These are some pictures of Glochidia larvae. They are kind of basically little clam-shaped things that latch on when near or sensing that they're in proximity to fish gills. They latch onto them, clamp on very tightly. They are parasitic. They are feeding on the blood and lymph of the fish through the gills, and like I said, stay on those gills for a few weeks to a few months. And some mussels have developed lures to lure in their host fish. This is a still picture from the Grand River I took a few weeks ago. The right in the center are the swollen gill filaments on this pocketbook mussel. And the lure, which I'll show you a moving picture of in just a second, you see there's a little eye spot there towards the right and what looks like a lateral line and fins trailing off to the left. This is what it looks like. This is head on. Same species, plain pocketbook. You see the eye spot, and when it starts to run, you'll see the swollen gill filaments between those two little fish-like lures. Oh, come on, you worked 10 minutes ago. Uh-oh, I lost it. Sorry, everybody. There we go. We're back. All right. Well, it doesn't seem to be working. But those flash back and forth, waving back and forth in the water column, luring in fish of all sorts.
You need to reshare your screen.
Thank you. Oh. Everybody see it now?
There you go.
Yeah, okay. I don't know if this video will work either. This is a, maybe. This is another species from Michigan, the black sand shell. You see the swollen gill filaments that look like teeth in the edge of the shell there, and that waving. I don't know what that's supposed to look like. It doesn't particularly look like a fish or an aquatic insect, but it does lure in fish who come in, and if they start to nip at it, they kind of invariably also puncture those swollen gill filaments and get the Glochidia larva onto their gills. And a very specialized one, I hope this video works, the snuffbox mussel, a federally endangered species that is here in Michigan. You see in this little picture some right here, some gill filaments that are swollen. It lives on the river bottom, and it has one known host fish, the log perch. which has a habit of moving along the riverbed, rolling over stones looking for aquatic insects. Periodically, they'll nose into a snuff box. Yeah, looks like this is going to work. Yeah. They'll bump that. Log perch will be along in a minute. And get caught. The female snuff box latches onto the nose of the log perch and kind of injects its larva out through the mouth of the log perch onto its gills. There's a wide variety of fish that are used by mussels as host fish. Some mussel species are fish generalists, host fish generalists. The giant floater, a very common species in Michigan, is known to use 24 different fish species as host fish. The snuffbox mussel that I just showed you is known to only have one, the log perch. And it is not a coincidence that the most common species around the state and the country and the world tend to be the ones that are host fish generalists. They can reproduce quite readily with a variety of fish. The host fish specialists are often pretty rare. As I said earlier, the snuffbox is federally endangered. Mussels are in trouble around the country, around the world. Dams interfere with the movement of the host fish. They also create reservoirs, which are very simple habitats compared to the rivers that mussels more typically live in. And so while you can have immense numbers of mussels in lakes and reservoirs, you tend to have a lower diversity, maybe only 1 to 4 species. Channelization, dredging channels, dredging streams, of course, is very, very bad on mussels. Poor water quality historically was a very big problem, less so now. When I started, we had a lot of problems with low dissolved oxygen around much of the state. That's largely been solved with improvements to wastewater treatment plants. Altered stream hydrology. This is a common problem in urban areas where you have a lot of impervious pavement. Rainfall hits that pavement, rushes into the river rather than percolating down through the soil, and consequently the stream is very, very flashy. It increases in flow volume and velocity very, very quickly and then comes back down. That is very erosive, and it destabilizes the stream beds, eliminating mussels. And zebra mussels, of course, are a gigantic problem that we're not likely to ever recover from. There are 6 federally endangered species in Michigan. 3 of them here in red have not been found in the state in at least 50 years. The scale shell hasn't been found in about 100 years. They are most likely extirpated, but the federal Fish and Wildlife Service still lists them as potentially occurring in Michigan. 3 others are still found in Michigan, the snuffbox being the most widespread. That little map on the right shows its range from that red line to the south, at least its historic range. The state, we have a larger list. Over 60% of the species in the state are on the list in some way or another, mostly special concern and threatened. Do we have any questions on mussel ecology before I go on?
Yes. One question from Abby. She asked, are the calcium carbonate shells of mussels at risk of deterioration like the coral reefs are as CO2 levels rise?
That's a very good question. I've not ever seen any studies of that. Certainly in the parts of the state, like the northern lower peninsula and particularly the UP, where coniferous forests are common, the spring runoff is often slightly acidic. And so the shells, the upper portion of the shell that's sticking out of the sediments tends to be what we call eroded. It is slowly dissolving, and consequently you seldom find mussels up north older than 9 or 10 years old because the shell has eroded to the point after that period of time that it perforates, and that quickly kills the mussel and opens it up to infection. I haven't seen any information on Increasing. Yeah, are we seeing decreasing pH around the state due to climate change? I've never seen any study on that. Most of the southern lower peninsula, I would think not. Our streams and lakes are pretty well buffered by the underlying geology. But up north, that's a possibility. That's a good question, particularly someplace like Isle Royale or the Keweenaw Peninsula, where you have a lot of basaltic bedrock very close to the surface. Good question.
Yeah, it is. It is. Here's another one that came in from Joyce. She's curious about that snuff box clam. How long does the snuff box clam hold its fish prey to transmit its infusion of larvae? And do the larvae in the gills kill the fish?
They've been known to hang on to the fish, and this is observed in laboratory studies more than anything else, for maybe up to a half an hour. The larvae are not, they're never known to have killed fish, and a fish that is infected once early in its life develops an immunity to subsequent infections, and so though a log perch might be captured 2 years later, the glochidia will not successfully attach to the gills and cause any problem.
All right, interesting. And then Jason wants to know if there are any fish species that host more mussels, maybe more than one species of mussel.
Oh, yes. Yeah, let's see, the minnows, the basses, the catfish too, I suppose, are host fish to, in this state, 15 or 20 different species of mussels, probably.
All right. Well, we're all caught up on those questions. So
Hi, Jason.
yeah, we can keep going.
All right. Because mussels are fairly rare, and some species very, very rare around the state, we have developed 3 different sampling protocols, we being a combination of DNR, Fish and Wildlife Service, some input from Eagle, formerly DEQ, and a couple of retirees, including me. We have 3 protocols intended to require mussel sampling and, if necessary, relocation in the footprint of certain construction projects. The river construction projects are things like bridge crossings, pipelines, things that will cross a river. Lake construction projects are maybe seawalls or boat ramps or dredging, which is kind of uncommon. And reservoir drawdown, that's a specialty thing all by itself. In the course of writing these protocols, we had to deal with some myths that surprisingly some of our colleagues believed. Mussels only occur in riffles, so we don't have to worry about them elsewhere in rivers. That's not true at all. Multiple species do very well in sand or silt. No, you don't find any mussels in canals, ditches, lakes, or reservoirs. Entirely untrue. Large lakes or reservoirs can contain millions of mussels. Several listed species occur in lakes, including the snuffbox. And in ditches, surprisingly, if they haven't been dredged in 10 or 15 or 20 years, you can have hundreds and hundreds and hundreds of mussels very densely packed. We also had to overcome the myth of old data. We have data in mussel occurrence maintained by the University of Michigan dating back to the 1920s at least. And sometimes people will say, well, there's been no mussels recorded in my area, my construction site for decades, or the most recent ones were found decades earlier, so they can't still be there. Well, that's not true at all. An example here is the Rouge River. The state-listed Lilliput mussel was found just last year. We found it 100 years after it was last seen alive in the watershed. So the fact that you don't have recent data for your project site doesn't mean that they aren't there. So the river protocol, it's applicable to fairly small scale projects up to a few thousand feet. Like I said, bridges, culverts, pipeline crossings. We've seen it applied, unfortunately, to large scale projects. Oil spills, giant dredging projects, large dam removals. It was never intended for that. There are other much more appropriate sampling protocols for those types of projects, and I think we've got everybody on board with not approving the use of this protocol for large scale projects. A typical bridge construction project would require the survey and, if necessary, relocation of mussels from 3 zones around the project if you were replacing this bridge, say, or doing significant repairs. The area of direct impact would be the footprint of the bridge itself. That might be removed and reconstructed or whatever. We also require survey and relocation from an upstream buffer, usually do that. Especially if you're going to put heavy metal excavators or backhoes into the river and work as you do the bridge construction work. And we also require surveys and relocations of mussels from a downstream buffer, typically a little wider, a little longer than the upstream buffer. That is to reflect the potential for sediment resuspension and burying of mussels with the construction activities in the area of direct impact in the upstream buffer. Lake and Reservoir Protocol has similar area of direct impact and buffer requirements. The lower little figure there might be a seawall that you'd put in, and the area of direct impact, particularly if you were putting it in, installing it with an excavator or some heavy equipment in the water, and then a buffer around it. The upper figure would be comparable to a dredging project, where the area to be dredged would be the area of direct impact, and we'd have a buffer surrounding it of some dimension to reflect resuspension and settling of sediment. The river and lake protocols are required to be used if you have state or federally listed species known or suspected at your project site, and you can only do so much, or can't do anything more to reduce the impact of your project. You've reduced the project footprint. You could get out of using these protocols if you move the project away from the mussels. That's more easy in river projects perhaps than in lake projects. An example would be installing a free span bridge rather than using bridge pilings in the river. It's required when you have state or federally endangered species. It is recommended for all other species, with the idea of trying to keep the common species common. We're intermittently, the state and Fish and Wildlife Service rewrites the list of endangered species, threatened species, and we'd like to not ever add to that list going forward. In practice, typically you'll do a reconnaissance survey to see if mussels are present in your area of direct impact and the appropriate buffers. And if so, you'll collect them from the ADI and the buffers and relocate them as necessary. The protocols describe in detail how to sample mussels, how to handle them, the data you have to collect to document what's there, and the procedures to relocate mussels out of the footprint of the project, out of danger, typically upstream or downstream at a very similar habitat. Mussel sampling is accomplished in shallow waters, which is to say like knee deep or so, by wading or snorkeling, and snorkeling being the better of the two, and in deeper waters by scuba diving. And sometimes mussels are pretty easy to find. This is a giant floater on the bottom of a lake in the Keweenaw Peninsula. This one's a little harder to see. All you see are the siphons, the filtration apertures that the mussel has. Otherwise, it's blending into that stream bed pretty well. Sometimes they're very hard to see. This one is, looks very much like the surrounding gravel and cobble. And sometimes they're almost impossible. They're totally buried. All you see are the little siphon apertures. It's right there. So usually when you're doing the sampling, you're not just looking at the surface of the sediments. You're turning, we call it raccooning, turning over the sediments with your fingers as you wade or move or snorkel or dive usually along transects across your area of direct impact or buffer zones. Which mussels to move? Again, kind of similar to when you have to use the protocol. You're required to move all state or federally listed species. It's recommended, again, that you move the non-listed common species too, again, to keep common species common. The reservoir drawdown protocol, the third protocol, that's kind of a different animal. Mussels react to drawn down water levels in different ways, and not consistently. They don't always move, quote, down slope, down towards the retreating water. Some mussels, apparently some species, tend to bury themselves and try to ride it out. Some will bury themselves for a couple of days, and if the water doesn't come back, they will surface and try to move. Just try to move. Typically, they move in random directions, not down towards the water, so that doesn't help. And a few species do start to move right away, but again, they move in random directions most typically. So reservoir drawdowns are hard. The mussels will survive a drawdown anywhere from a couple of days to a couple of weeks, depending on the temperature, air temperature, the moisture content of the sediment around them, and on species too. Some seem to be more resistant to drying out than others. But a couple of days to a couple of weeks, that's a very short period of time. A lot of reservoirs are so big, you have almost no chance of, say, walking the perimeter to try to find and collect all the mussels you can and move them someplace, probably upstream of the reservoir. And especially if the reservoir is full of very soft, silty sediments, it may not be safe to walk out onto that former lake bed. For people, they'll sink to their hips and it'll be very hard to get out. So reservoir drawdown. We're learning a lot about reservoir drawdowns, and DNR, I think, is quite flexible in recognizing the reality of the difficulty of surveying an entire reservoir for mussels. We're funding some research, which is to say DNR and a couple of power companies up in the Upper Peninsula. Lake Superior State is looking into where mussels occur in reservoirs to see if we can come up with some predictive patterns that we might use to direct people trying to salvage, rescue mussels on the bottoms of reservoirs. And it remains to be seen if there are any predictive variables like sediment composition, lake bed slope, presence or absence of macrophyte beds that will allow us to have people doing salvage operations in a better focused manner than they do now. Survey timing. Of course, specific dates will be determined by the project schedule, but in general, we really like people to work with mussels between June 1st and October 15th. That's when the water temperature is warm enough that mussels will be near the surface. In the winter, a large majority of mussels will bury themselves, not deeply, a few inches, but they'll be much harder to see and salvage if you go out when the water is still very cold. Also, if you are relocating the mussels, the relatively warm water from June 1st to October 15th makes it more likely that the mussels will survive. They will be able to rebury themselves appropriately and make it through the following winter. And the moderate air temperatures as you're handling mussels, identifying them, measuring them, having them out of the water is just less stress. In general, to minimize the requirement of mussel surveys and relocations, there's just some examples. You build a clear span bridge that does not have any pilings or piers in the water. You install a seawall from the lake shore or the river shore, the riverbank, rather than putting heavy equipment, excavators or whatever in the water. Reservoir drawdowns, you minimize the reservoir drawdown depth if at all possible. If you're repairing a dam and you only need to take down the water 3 feet, do that. Don't draw down the water or the reservoir in its entirety. And any project, keep the big equipment out of the water. Questions on the sampling protocols?
Yeah, we do. Rex asked, how prevalent is the eastern pond mussel in Northeast Michigan?
Pretty common. No, let me take that back. That's an unusual mussel species. It does occur mostly in northeast Lower Peninsula, and there's a couple populations in Lake St. Clair, I think. It is not a common mussel. It's a listed mussel. I wish I had my list of species. It is a state listed species. I don't think it's endangered, but it is perhaps threatened. So yeah, it is not. It is not a common mussel.
Thanks. And then we had a set of questions that came in when you were discussing drawdowns and their impacts on there. And a really great question from Abby: how do mussels move?
Mussels move. I should show. Gosh, that's too bad. Well, to take a look at the photograph there on this slide. That's my hand there. The left side of both those mussels, which are plain pocketbooks, they're inside the shell is a flap of tissue called a foot. It can extrude outside the shell itself when the shell is open slightly, and mussels move by moving that foot further into the sediment and pulling the shell back towards the foot. It's a contraction thing, extension and contraction. And of course, these mussels in the sediment would not be oriented like they're in my hand. They would actually be tipped up to the upper right. And so they extend that foot, pull themselves up against it, extend the foot again, pull themselves up against it. Seasonally, mussels don't move too much. Some barely move a few feet in their lifetimes. In a drawdown situation, they're capable of motoring along at I've seen mussel trails in drawn down reservoirs that were 30 feet long. I don't know how long it took them to move that far, but you can see them slowly moving. If they move 24 hours a day, they could move probably 20 feet in a day if they were motivated to do so.
All right, that's interesting. I think Abby posted a YouTube link in the chat. I'm guessing that's what it is of
cool.
Mussels moving.
Good.
So another question from Rex: what relocation protocols are used after a dam fails?
You would use the drawdown protocol, and if you see a drawdown, starting with drawdowns, if you see a drawdown coming, that gives you time to get out in the local area and identify a good place to put the mussels, as I say, typically upstream, maybe downstream in certain circumstances. In a dam failure, you don't have that time. Dam failures happen too quickly, of course, and unexpectedly usually. And so it would be an emergency situation. A dam failure very well might release a lot of sediments downstream, creating unfavorable conditions to move mussels to and for the resident mussels. So your relocation site probably would not be downstream. It would probably be upstream of the reservoir. And you can find a relocation site in a few hours with a little luck. So you would have time to scout that out while simultaneously starting to collect the mussels and move them out of the footprint of the reservoir.
All right, thanks. Jason asks, is there an anticipated date for completion of that reservoir research project that you mentioned?
We've done 2 field seasons, and next year is the third and last. And I don't know when Lake Superior State owes a report to the power company and DNR. I would guess probably by the middle of the following year, so 2024. There's another project coming, or another opportunity to collect this kind of data. DNR just received a large grant from the National Fish and Wildlife Foundation to remove 17 or 20 dams around the state, mostly small dams, mostly owned by DNR, and so they'll be, that'll be a controlled situation. A high end, high sample number. Hopefully, I was just discussing that with DNR this morning. Hopefully, we can incorporate some quick qualitative, semi-quantitative studies of where the mussels are found in those 17 or 20 reservoirs and really learn a lot about whether it's at all possible to predict where mussels will be in reservoirs. So But yeah, the Lake State study will probably have our final report out sometime in 2024.
All right. And then one more question on protocols. Amani asks, what criteria would you use to determine if a day has good conditions for a survey, perhaps flow rate or water level?
In rivers, the protocol actually the lake protocol too, discusses water clarity recommendations. I mean, you can find mussels totally by feel. I've done that many times working in turbid lakes and rivers. You're better off if you can see what's going on. So it contains a recommendation on water depth of transparency. I don't remember what the number is, but it's something on the order of at least a foot. So if you're especially if you're diving or snorkeling and your face is close to the stream bed or the lake bed, you can see what's going on. Flow in rivers, that kind of relates to survey safety. And I don't recall that the protocol has a recommendation for that other than the blanket statement that if it's moving too fast for you to stand up or hold your position snorkeling or scuba diving, it's not a good day to be out there.
All right, that makes sense. Thanks, Joe. I think we've caught up on the questions.
All right, thank you. So, identification, like I said, there's 42 species found or previously found in the state. And until recently, we did not have any identification guidance. So a few years ago, Renee Mulcrone and myself wrote a field guide to the mussels of Michigan with photographs and range maps and all that kind of stuff. And simultaneously, the Michigan Natural Features Inventory published a very, very lovely poster of the freshwater mussels of Michigan. If we'd met face to face today, I was going to hand these out to you, as you might be interested. Alternatively, you can, as the bottom of the slide says, you can email me for PDFs of these documents, or in a few slides, I'm going to give you the email addresses of the two people who are responsible for mailing out the field guide and the poster. So the field guide, this is a sample page. We have photographs of the shells. Almost all the shells taken from the University of Michigan collection, a few from the private collections of Renee and myself. We have a shell description there in very abbreviated terms. We have typical habitats where they're found, though you always want to take that with a grain of salt. The listing status, is it federally listed, state listed, not listed? Similar species, that's handy. Other things that might look at, so you can look at those pages. And a range map, and that's interesting. We've been updating the range maps every winter. I send out an email to the 30 or so mussel heads around the state that I am in contact with, asking for updates to the range maps based on that year's field work. And the first year I did that, we got about 3 dozen updates. And the last couple of years, we've been getting about 10 updates each. We hope to have updated range maps every year, probably on a University of Michigan website. And so if you're really interested in that kind of thing, drop me an email and I'll let you know when we set that up. Renee and I hope to work on that this winter. Just for general interest, the most common and rarest species in Michigan, the most common species by far are probably the giant floater and the fat mucket. The fat mucket is interesting. It's sexually dimorphic. The male and female look different, sufficiently different that if you didn't know they were the same species, you might think they were a different species. The rarest mussel in Michigan is the club shell, which is found only down along the southern border of Michigan and Ohio, in the St. Joe River that's down there, not the big St. Joe that's in southwest Michigan. The little St. Joe over there flows into the Maumee eventually. There are probably a few hundred of those still living in the St. Joe, the club shell. IDing mussels is fairly challenging. Between species, there's limited shell shapes. They're all kind of oval or maybe round. Limited color variation. They're all tan, brown, or black. Juveniles are very, very hard. We won't go into that. And within a species, it's they can be surprisingly variable in shape, say from headwaters to lower down in the watershed. There are fairly characteristic gradations of shell shape within a single species that you can expect to find. And of course, some species are sexually dimorphic, like the fat muckets here. Like I said, if you didn't know that, you might think that they were separate species, different species. The field guide, where appropriate, illustrates the male and females of the sexually dimorphic species. There's 10 or 11 of them in the state that have significant sexual dimorphism. You use the shell to identify the mussel. And I won't go into this, but there are various features on the outside of the shell and the inside of the shell that, if you total them up, can guide you in a good direction as to what species you might be dealing with. When I teach mussel identification workshops, I discuss 11 different features of the shell. And fortunately, 9 of the 11 are on the outside of the shell. So you don't need a dead specimen to identify most species. If you're interested in mussels and you need photo ID help, or you need help with identification, you can send me pictures. The major mussel heads around the state get pictures every year from various folks, watershed groups, and so on. If you do that, please take at least 2 photos. One is a kind of a profile shot, like there on the left, with anything in it for scale. Hold it in your hand, put a penny next to it, put a ruler next to it. Anything will do, but it's good to get the scale. If you have a dead shell, photograph the interior of the shell so we can look at the teeth that are up along the ventral margin of the shell and identify. That helps a lot in identification. And it's also useful, whether you're dealing with a live or dead shell, to photograph it from the top so we can see how thin or expanded, excuse me, compressed or inflated in our terms, the shell is. And also get a feel for the details of the ridges and such that are up by the beak, which is here. You can see my arrow. Send us those 3 photographs, and we have a shot at identifying the species. Excuse me. Training opportunities. In the last 14 years, I've taught 25 ID workshops for about 440 people. I'm starting to wind down on that. The most recent major workshop was sponsored by the Michigan chapter of the American Fisheries Society. We did that in Grand Rapids in August. We're likely to repeat that in 2023, probably again in August. And we do them elsewhere too, at universities or for watershed groups sometimes. So drop me a line if you want to talk about doing an ID workshop. I'll probably do a few more of those before I totally step away from them. I'll take questions on IDs, and while Joe reads them to me, if you have any, this is where you get the field guide and the poster. The field guide comes from Jessica Pruden from the Fish and Wildlife Service, and the poster comes from Pete Badra of the Michigan Natural Features Inventory. So, Joe?
Well, Joe, we actually didn't have additional questions come in about ID.
That's fine. That's fine. I'll leave that up for a few more seconds, and then we'll move on to the last part of this talk. All right. If you want to work with mussels yourselves, do your own identifications, do your own surveys, do your own relocations. It's totally possible. It takes time and experience. The survey protocols are relatively simple to follow, particularly for smaller projects. The identification, which of course is part of it all, that takes more experience. 40-some species in the state, different sizes, sometimes different sexes. There's no getting around. It takes experience. So if you want to get good at identification, attend the workshops whenever they're available. Start your own shell collection, although that requires a permit that I'll discuss in a minute. least hypothetically requires a permit. And you can make if I send you the PDF of the field guide, you can make your own regional or watershed specific field guides. So you're dealing with just the whatever, 8, 10, 15 species in your watershed rather than the book with all 40 plus. And this is an example of that. I put this together for the Maumee River. I took the photographs and a summary of some of the significant shell characteristics from the field guide, cutting and snipping the photos, put them into a Word document. It was only 6 pages long for the 18 species that are known to occur in the Menominee, in the Upper Peninsula. So if you're a watershed group and you want to spend a little bit of time doing this, that's real easy. I'll send you the PDF to the field guide and you can do that. Permit requirements. If you're going to work with mussels, you have to have at least one permit. The scientific collector's permit, which you get from DNR Fisheries Division. If you need these permits, please drop me a line and I will I'll send you the contact information. If you're going to work with state and endangered species, threatened and endangered species, you get that permit from the DNR Wildlife Division, interestingly. If you're going to work in waters that may have federally listed species, which again is only the southern lower peninsula, you have to get a permit from the Fish and Wildlife Service. The 2 state permits, their turnaround is usually pretty quick. The U.S. Fish and Wildlife Service, you have to prove a lot of things to them before they'll give you a permit. It can take over a year to get a permit, sometimes more than, sometimes 2. And then finally, the shell ID test is soon to be part of the state permits. We've developed an identification test using shells from the species around the state intended for consulting firms and other for-profit organizations that do mussel surveys and relocation projects. It is not required for universities, for watershed groups, or anybody like that, just for the for-profit consultants. We have the test put together. It will be recommended to consultants for the field season of 2023, and starting in 2024, it will be required for any project conducted by a for-profit company. Almost final here, I guess. Some state of Michigan mussel heads that if you know them or you want to contact them, I can get you their contact information. Peter Badra at the Michigan Natural Features Inventory at Michigan State. Several people in DNR: Jen Johnson, Cleo Harris, Patrick Ertel, Ellie Galati, Kesare O'Brien. 2 people at Eagle, Amanda Chambers and John Matusik. 2 people at MDOT, Jeff Markowitz and Dave Dortman, and a variety of university folks. The prominent mussel research universities in Michigan are Central Michigan, Lake Superior State, and just recently Grand Valley State. They've started doing some work with mussels. Those folks are, I've always found them to be quite cooperative. And if you send me a question that I can't answer, I will refer you to the appropriate person on this list probably. There are also a variety of consultants who work in Michigan. I don't want to go into them because I don't want to, recommend anybody over at anybody else. But I can send you a list of the the major ones if you need that kind of information for some project you have. There again is my email address. So if you want to contact me, that's where I'm at. So any questions?
Thanks, Joe. That was really great. I know I always learn something new when I'm listening to you talk about mussels. There's always something new to learn. Again, I'll remind people, if you have questions for Joe, he's done with his slides here. So if you have additional questions about mussels, you can add them to the chat at this point. I also typed into the chat Joe's email address and also the email addresses for Jessica and Pete that he shared if you're interested in getting copies of the field guide or the poster. I know I have both. I use them both. The field guide is really handy. It's really nicely done, and you can just get it. It's great. Like, they just give it to you. So that's amazing that we have that resource for us. So you have this opportunity now to pick Joe's brain with any other mussel questions that you have. And here comes one now. Josh asks, "Hi, Joe, how can someone interested sign up to take that mussel test?"
Yeah, good. Contact, I would say Jennifer Johnson at Michigan DNR. If you don't have her email address, email me and I'll put you in touch with her.
All right, next question from Amani. How can mussel survey data be evaluated? Like macroinvertebrates have a water quality scoring index.
Yeah, the back up. Amanda Chambers with Eagle has talked about trying to develop something like that for some years. It's kind of hard for a variety of reasons. Macroinvertebrates are more widely distributed than mussels. Mussels are very, very patchy, even in optimal habitat. We don't know why they aren't more consistently distributed in what seems to be prime habitat for mussels, which tells you probably that we don't really have a good idea what is prime habitat for mussels. There's also the complicating factor of the host fish. You can have lovely habitat, but if, say, a dam is preventing the host fish from moving upstream and kind of seeding the riverbed with juvenile mussels, they won't be there. So if you do have mussels, that tells you multiple good things about the river or the lake. The dissolved oxygen is sufficient. There are not significant concentration of toxic chemicals like ammonia or copper. The hydrology is pretty good. The host fish are present. But if the mussels are missing, that doesn't necessarily tell you anything. You don't know why. With the macroinvertebrates, it's assumed to be poor water quality or perhaps hydrology. It's not that simple with mussels. There are more reasons they might be missing from a particular location, especially if your sampled site is fairly small. If it's only a few hundred feet long, they could just be naturally missing or lacking for reasons we just don't understand.
All right, Rex asked, you mentioned copper. At what levels does copper become hazardous to our mussels?
Rex, send me an email. I'll have to look up that number.
Yeah, good question. Good point. Yes, thank you. Are zebra mussels the only invasive mussel species in Michigan?
No, there's the very similar quagga mussel. Those are the only two so far. Elsewhere in the country along the Northeast, there's the green-lipped mussel, which is typically an estuarine species elsewhere in the world, but seems to be invading freshwater systems. I'm sorry. There's also the Asiatic clam, Asian clam, Corbicula, relatively small, about the size of your thumbnail, but terrifically prolific. You can find in a square meter in good habitat, you'll find hundreds of them, including and thousands of their shells. Is quite remarkable. The evidence, it's very clear that zebra mussels and quagga mussels are devastating to our native mussels. Corbicula has been in the country for over 100 years, and the evidence, the studies are mixed as to whether even dense populations of Corbicula have much impact on the native mussels. I have certainly found native mussels in what I think are fairly healthy populations in situations where I find dozens to a low, maybe 100 Corbicula mussels per square meter. So.
Yeah, I know I was out surveying in the Red Cedar River with some undergrads earlier this fall and was shocked at the level of Asiatic clams in the river. I did my dissertation research in that river, 15 years ago, and they were there, but they weren't in the numbers that they are now. It was pretty amazing between the shells and the live clams. A really good question from Josh. Can you explain the difference between a mussel and a clam?
I usually lead off with that information. I'm a marine biologist originally, and in the marine world, there's a difference between clams and mussels. This is kind of a complicated answer. Clams live like our native mussels do. They live on the bottom, free living, not attached to any hard substrate. Whereas marine mussels, like oysters and things, or zebra mussels, are attached to rocks and pilings and even ropes. So we should call our free living, on the bottom, not attached, bivalves, in freshwater clams. For some reason, we call them mussels. Most people in the profession use those terms interchangeably. The literature usually uses mussels. And also, ironically, as near as we can tell, our freshwater bivalve fauna evolved from marine clams, not marine mussels. And so about 300 million years ago. So the short answer is we use, in the freshwater world, we use those terms interchangeably.
Great answer, though not a simple one.
No.
And so I have a question for you. So we have, a lot of the people at this conference are involved with volunteer stream monitoring. They're out using, kick nets to collect macroinvertebrates, and invariably they're often going to run into having mussels in their nets. How should they handle them?
Let's see. Let me back up to a good picture of a clamshell. Okay, that'll do. You can photograph them if you like, and send me the photos if you want to identify them. But if they're alive, you should put them back in the stream bed. And you see these photographs. You see the shells. At the very top, there's a little bump. We call that the beak.
And we can see your cursor if you want to use your arrow.
Okay, thank you.
Yep.
So that's the beak. That's the beak. That's the beak. That's the beak. And you see that in these, especially in the club shell, but even all of them, if you drew a line in the very center of the shell, the shells are not symmetrical. The beak is offset a touch. That results in a comparatively short end behind the beak and a fairly long end in front of the beak. That long end, when you put the mussel back into the sediments, the long end goes up. This end, etc. The long end goes up. It's really important that you put them back that way if you choose to plant them. You plant them kind of like a tulip bulb. You can stick them in about halfway. You can bury them all the way if you like, but typically you would just stick them in about halfway. If you're not sure which end should go up, just set them on the riverbed or the lake bed and let them burrow in themselves. They can almost always do that in fairly short order. It's much better to do that than to put them in upside down with the long end down. And that takes an immense amount of energy for a mussel to reorient itself. And some of them may not be able to, and they'll just die. So long end goes up.
All right, thank you. I appreciate that. A question that I get sometimes, I think I have a little bit of knowledge about it, but I'm really interested to hear from you. I get questions from our lake residents and volunteers. A lot of them see zebra mussels in their lakes, and they've seen, either those zebra mussels attached to native mussels or, just seen a general decline in native mussels in their lakes. And, there's a lot we could talk about that. But now, they're hearing about quagga mussels, as you mentioned, another invasive species related to zebras. Are we seeing those in our inland lakes, or is that more of a Great Lakes problem?
Last I knew, that was more of a Great Lakes problem, but you folks have a better feel for that than I do. Yeah. Last I knew, it was primarily a Great Lakes problem, and the connecting the connecting channels between the Great Lakes, the St. Clair River, Detroit River.
Sure. Yep. All right. We have a question that just came in from Rex. Are any of our native Michigan mussels safe for human consumption?
They all would be, as long as they are not collected from a place that has high contaminant concentrations like metals or PCBs or something like that, which most water bodies do not have that. I've never eaten one. I'm told they're quite tough. They're quite bland, and if you're steaming them, you have to steam them for a very long period of time before they soften up enough that you'd care to chew them. But indigenous peoples, Native Americans, ate them quite regularly. And if you get a big mussel, it's as big as your hand, and the flesh inside is a half a pound or so. So you don't have to collect many of those to get a good meal.
All right, that's great. I was going to follow up if you didn't answer whether you had ever tried one before or not. So I'm glad that you jumped right there to that answer. It looks like we've gotten through all of our questions. We'll hang out for a moment to see if anybody else has any, questions they're thinking about, or they're trying to type it in, or if you'd rather just unmute yourself and ask a question of Joe while we have him here, feel free to do that. But I will say, again, Joe, thank you so much for sharing this information with us. These are fascinating animals, ones that we don't, maybe notice as much as some of the more active swimming critters or the big waving plants that we encounter. But, I think they're definitely a fascinating thing. We do have a comment from Abby. She has a new appreciation for mussels, so thank you. And then we have a question from Eric and Georgina. How destructive is wave energy released by like wake boats to mussel beds? Do you have a sense of that, Joe?
Yeah, waves in general, usually in lakes, usually exclude mussels from the very shallowest margin of the lake up by the shoreline. Just natural wave action does that, to the tune of, you seldom find them in less than a foot or two of water. And if wakes are common in lakes or natural waves, they are capable of moving into deeper water, slightly deeper water, to get away from the wave zone. It's kind of common to find decent numbers of mussels in lakes on the upwind side. Our dominant winds in Michigan are from the west, and often you'll find higher mussel numbers on the west side, the north side, the south side, rather than on the open east side where wave action is disruptive to them. The problem is probably that their tiny little juvenile mussels, when they drop off the host fish, can't settle efficiently in places where there's lots and lots of waves.
Right. Great. Interesting. I know that's an issue of, these wake boats and other, high energy boating that's happening on a lot of our lakes is raising a lot of questions and a lot of concerns right now across Michigan. So
it certainly should. And it'd be disruptive to the sediments too. If you have soft, mobile, resuspendable sediments that mussels might like to be in, if they're constantly being disturbed and resuspended, the juveniles will not be able to survive there, and therefore you won't have adults.
Right. That makes sense. Now that we know a bit about mussel ecology, it makes sense. All right, Joe, I think we're going to wrap up this session. Thank you so much again for coming, and all of you in the audience. We appreciate you being here. We're rolling into our lunch break now. Lunch runs from noon to 1. You're on your own. Of course, we're sad we can't feed you and have a meal with you today, but we'll on break from noon to 1. And then at 1 o'clock, the afternoon workshops will begin. You have the choice of stream macroinvertebrate session with Paul and Tamara, or lake data interpretation with Eric Elgin and myself, and that will start at 1 o'clock. Look at your agenda to find the Zoom links for those, and we will see you there.